Today, we dive into groundbreaking research published in NPJ Regenerative Medicine that explores how rejuvenating blood stem cells can extend the lifespan of old mice. The title of the study is Transplanting Rejuvenated Blood Stem Cells Extends Lifespan of Aged Immunocompromised Mice. This study by Carolina Florian from the Belvich Institute for Biomedical Research in Barcelona, Spain, and other collaborators sheds light on an exciting frontier in regenerative medicine. Aging is often accompanied by a decline in the regenerative capacity of stem cells. This research aimed to restore this capacity using a specific inhibitor called casin, targeting the small Rho GTPase CDC42. A key point of this study is that rejuvenating hematopoietic stem cells in some way, and then transplanting these rejuvenated cells into older mice, helps rejuvenate the mice and extend their lifespan. The paper states, the hematopoietic system is the carrier for many rejuvenation factors, and this leaves open the possibility that rejuvenating aged HSCs might represent an effective strategy to improve aging of the whole organism. But why CDC42? Researchers have long been interested in CDC42 due to its crucial role in various cellular processes, including cell polarity, cytoskeleton organization, and cell cycle progression. CDC42 is essential for maintaining the proper function and regenerative capacity of stem cells. Increased levels of active CDC42, CDC42-GTP, are associated with aging-related phenotypes and impaired stem cell function. As one study noted, constitutive activation of CDC42 in mice results in a premature aging-like phenotype affecting multiple organs and overall fitness. This led researchers to hypothesize that inhibiting CDC42 could counteract these aging-related effects. Casin, or CDC42 activity-specific inhibitor, is a small molecule inhibitor designed to target and inhibit CDC42. By reducing CDC42 activity, casin helps modulate these processes, particularly affecting the regenerative capacity and aging-related functions of stem cells. In this study, aged mice were given a short systemic treatment with casin. This treatment led to significant improvements in their fitness, endurance, and strength, comparable to younger mice. One notable quote from the paper states, casin treatment overall improved greater than 2-fold mouse endurance and 1.5-fold grip strength compared to aged control mice. The researchers discovered that casin treatment increased the regenerative potential of aged skeletal muscle stem cells. By reducing CDC42 activity, they observed enhanced myogenic colony capacity and divisional kinetics in aged stem cells, resembling those of younger mice. Decreasing CD42 activity in aged muscle stem cells by ex vivo casin treatment, already at 0.5 micromolar doses, significantly increased divisional kinetics and myogenic colony capacity of aged stem cells to resemble young muscle stem cells, the study notes. But the effects of casin didn't stop at muscle cells. The treatment also increased hematopoietic stem cell HSC polarity and restored their localization in bone marrow. But what does increased stem cell polarity mean, and why is it beneficial? Stem cell polarity refers to the asymmetric organization of cellular components, which allows the cell to divide asymmetrically. This means one daughter cell remains a stem cell, self-renewal, and the other differentiates into a specialized cell type. Proper polarity ensures efficient and correct asymmetric cell divisions, maintaining a balance between self-renewal and differentiation. Increased polarity can enhance the regenerative capacity of stem cells by ensuring efficient asymmetric divisions, leading to a more effective replenishment of stem cells and production of differentiated cells needed for tissue repair and maintenance, the study explains. With aging, stem cells often lose their polarity, leading to symmetric divisions that either produce 2 differentiated cells or 2 stem cells. This disrupts the balance, resulting in either a depletion of the stem cell pool or insufficient differentiated cells for tissue maintenance. The loss of polarity is associated with changes in cell signaling, epigenetic modifications, and cytoskeletal alterations. By increasing stem cell polarity, treatments like casin can restore proper asymmetric divisions, enhancing the regenerative capacity of aged stem cells. This helps maintain tissue homeostasis and counteracts the effects of aging. Let's talk about the transplantation process. The researchers isolated HSCs from donor mice that had been treated with casein. These cells were then transplanted into aged immunocompromised mice, specifically KIT W41J mutant mice, or NBSGW mice, which lack a functioning immune system. This allows the recipient mice to accept the transplanted cells without the usual immune rejection. Normally, bone marrow transplantation requires irradiation to eliminate the recipient's existing bone marrow cells and make space for the new cells. However, in this study, irradiation was not required. The recipient mice were immunocompromised and could efficiently engraft the transplanted cells without the need for preconditioning treatments like irradiation. This made the process less invasive and less stressful for the recipient mice. The isolated HSCs from the donor mice were transplanted into the recipient mice via intravenous injection, allowing the transplanted HSCs to travel through the bloodstream and home to the bone marrow. The most compelling finding was that transplanting rejuvenated HSCs from casein-treated mice into aged, immunocompromised mice significantly extended their median and maximum lifespan. Aged mice receiving HSCs from aged casein-treated mice presented with a significant increase in median 24.8% and maximum plus 34.0% lifespan, the study highlights. Mice transplanted with aged control HSCs showed no changes in lifespan compared to non-transplanted mice. This study presents a promising strategy for rejuvenating aged tissues and extending lifespan by targeting endogenous stem cells. By demonstrating that rejuvenated HSCs can have a systemic rejuvenating effect, it opens up possibilities for various types of rejuvenative treatments on HSCs to have similar beneficial effects. The findings highlight the potential of stem cell rejuvenation in improving health span and lifespan. If you found this research as fascinating as we did, don't forget to like, share, and subscribe for more updates on the latest scientific breakthroughs. Stay curious, stay young. Until next time, embrace the future of aging.